We begin with a regular strand of DNA laid out flat. Our legend indicates which nucleotides are which and what bases are to go along. We begin the process of DNA replication by splitting the DNA with a helicase enzyme. The helicase works its way across the middle of the DNA, separating the weak hydrogen bonds that hold the two strands together between the nucleotides that complement one another. Now, as shown, we have two separate strands, but they themselves are single-stranded. We have now created a replication fork as shown where the two strands cross over one another at the far right of the diagram. If we were to continue the splitting to make a larger shown piece of DNA, the helicase would continue to move 3' to 5' direction. The green balls on the left represent SSB, or single-stranded binding proteins. They help keep the two strands separated. We have now an opening, or a replication bubble, where we can begin the synthesis. RNA primase is the next enzyme that is to be used. After the strands have been separated, RNA primase begins the DNA replication by bringing in the complementary nucleotides to suit a certain number of original DNA bases. We have now our primer. Once completed, DNA polymerase, another necessary enzyme used in DNA replication, is brought to continue the new daughter strand running 5' to 3', and completes by attaching the complementary nucleotides to attach to the original strand. The arrow shows the 5' to 3' direction the new daughter strand runs in. The leading strand is now completed for the time being as it is all paired. The lagging strand comes next. Shown under the leading strand, we now have the lagging strand. Because of the opposite running or anti-parallel directions the strand run in, we create a similar, though different construction of DNA replication. RNA primase is what we use to start and essentially end the now called fragments we are going to make. Shown in the diagram, RNA primase is brought to add the corresponding nucleotides to the original strand. Once constructed, we build yet another sequence of nucleotides using RNA primase further down the strand. We now have two primers, though they are separated with a gap to be filled. DNA polymerase is brought and is going to build Okazaki fragments in the gap the primase created, this time running the opposite direction of the fork. The DNA polymerase delivers the nucleotides needed to make the fragments. Labeled in light blue, we have now successfully created an Okazaki fragment. Further down the strand again, RNA primase is brought to make another gap where an Okazaki fragment is to be made. Another Okazaki fragment has been built as the DNA polymerase filled where the gap. Though there appears to be a problem. Labeled in red, a cytosine has been added where an adenine was supposed to. DNA polymerase comes back and removes the cytosine and adds the adenine. The Okazaki fragment is now correct. For reference, once again, another Okazaki fragment is to be made past the third primase. We have now two new, yet same strands of DNA. The two new strands are known as semi-conservative, because one strand is new, yet one is the original. Finally, DNA ligase, another enzyme, is brought to run across the new strands to seal the fragments together.